All right, to start the award session, the winners of previous year's awards were invited to give a brief overview of their experience and update on their research. As many of you know, the VESS offers 3 awards each year. Information for these awards can be found on the website. The awards we offer are the VESS Travel Award. The primary purpose of this award is to provide the recipient with the opportunity to visit one or more vascular surgery centers of excellence. The visit is intended to stimulate academic inspiration and to foster development of fraternal fellowship in vascular surgery. The travel grant for 2022 was awarded to Dr. Jonathan Bath from the University of Missouri. Dr. Bath?
Thanks. So first of all, thank you to the Society for the invitation to present. These are my disclosures. So a really big thank you, first of all, to the VESS Grants and Scholarship Committee, Dr. Jason Lee, Dr. Ben Brooke, Dr. Veeraswamy, and Angela, obviously. I scoured the internet for a picture of Angela, and she is just not to be found anywhere, so congratulations on your anonymity. We've all had these kind of patients that come in essentially urgently. In this case, it was a ruptured juxtarenal aneurysm with a patient that was not fit for really anything at all, and certainly couldn't get an open operation. So a customized solution was made, and I show these slides only to really sort of demonstrate how complicated these can be from the point of view of radiation exposure, contrast exposure, and just complexity of the operation. In this particular case, this was a laser fenestrated approach. So this juxtarenal ruptured aneurysm in an obese patient underwent laser fenestration, a complex operation on an old hybrid machine. The radiation dose was high, the contrast dose was high, and this is just a complex operation all around. So this is a growing issue, clearly. It's not going away, it's only getting worse, and certainly for patients, this is a true phenomenon. Although seen rarely, I must admit, in our place, a lot of our cases often exceed 5 gray. I routinely get a warning that I have to go and check up on these patients afterwards, and perhaps that's something that we can improve upon, but it does happen routinely. Obviously, significant contrast dosing as well for these patients. They come in as an emergency and urgent presentations, get a CT scan, they might get a follow-up CT scan, re-interventions, and it's the perfect storm for significant kidney injury. For the operators themselves, this is a research letter that came out of England, and actually they took lymphocyte samples from 2 groups. You can see high-volume operators and those who are radiation naive. There's a significant number of chromosomal rearrangements to those patients, those endovascular operators with a significant radiation exposure. Whilst we don't know the clinical sort of significance of this, obviously this is worrisome. And everybody knows about Ted Dietrich, who finally died, I think, of a brain tumor, but certainly was a living example, in his own words, of what excessive radiation can do to tissue. So this really formed the thrust of some of my visit to the University of Pisa. You can see some of these great pictures here of electromagnetic navigation bronchoscopy. It really looks like the real thing. And you sort of realize that this is sort of the next level in the virtual environment that you can create. Here on the left-hand side, you can see the FALL system by Philips and a counterpoint, the Centerline IOPS navigation system, who are working existingly in this space. If you type into the search bar electromagnetic navigation, you can see on the left the number of papers have increased, although honestly speaking, you can type absolutely anything you want into the bar and you'll see this trend too, so don't put too much by that. But it's certainly an emerging technology, and we've looked at this at our own institution, the University of Missouri. The catheter array is a bit limited right now. The wire arrays too are tricky. The equipment has an expensive upfront cost, but certainly this will be the way of the future, and we really need to get in on this, I think. So I had the opportunity to go and visit the University of Pisa, and these folks here highlighted are the folks with whom I collaborated. You can see in 2012, this is when this paper was written, they were already in this space and doing this work. They have 3 modules to electromagnetic navigation. I'll go through what that is in a minute. Essentially an imaging module first, the sensorized instruments, and then a graphical user interface so you can see actually what you're doing in the aortic phantom. Sensorized tools are as simple as catheters and wires that essentially have electromagnetic sensors to allow them to be seen in a virtual environment. The team at the University of Pisa actually gone to the trouble of making some sterile sheaths that are sensorized as well as laser fibers. So they're sort of really pushing this along. And they actually have a fantastic testing paradigm for using electromagnetic navigation using laser fenestration. They've done some great experiments by 3D printing, essentially, synthetic replicas of patient anatomy. And you can see embedding these in the middle array in a foam pelvis so that they can actually do the work in the virtual environment. And they've actually done some accuracy testing as well. You can see down there on the left-hand side the actual model, and on the right the graphical user interface, or the aortic phantom, which allows them to track how accurate they are. So the University of Pisa, located there in the northwestern portion of Italy, has a research house, if you will, called EndoCAS, the Center for Computer-Assisted Surgery, with 3 arms, and they certainly do a lot of research in this zone. The leadership here, Professor Mauro Ferrari, which you can see on the top left, he is a vascular surgeon who's headed up the unit since 1991, and on the right side is its counterpart, the biomedical engineer. And this group really has a number of postdoctorate students as well as full-time PhDs that run the center. So this was a 3-day experience sort of at the University of Pisa. The first day, after sort of settling in, we met with the vascular surgery unit, toured their facilities, which looked like any other facility except the Pisano Mountains in the background. And then the highlight for me was really joining Case Conference, where they have a combined sort of cardiovascular, cardiothoracic experience with the vascular trainees. So it's one sort of allocentric unit, if you will. And that was really interesting. There wasn't sort of a back and forth as to you do some, we'll do some. They really do these cases together. After an extended dinner and a late wake-up the next day, we had this aortic summit, which was put on by Dr. Ferrari, sort of a mini aortic summit, which I really enjoyed. We had the opportunity to talk about some of our customized experiences, but Professor Giovanni Protesi from the University of Genoa was one of the highlights for me. This is somebody that I hadn't had knowledge of before. He's showing here a picture of his dedicated aortic sort of emergency helicopter system. They have obviously dedicated aortic beds, and they have a huge cardiothoracic and vascular unit that's combined. In Italy, they have a fantastic sort of registry, the multinational registry for all of these new devices that they get to use, and he's a member of a number of them, one of which is the inbreed registry, looking at inner branch devices and these devices we don't have access to here. So it's fantastic seeing his experience with these customized grafts. Day 3 was a tour of the EndoCAS facility, and I'd obviously done a lot of back-end work and Zoom conferences, etc., with my collaborators. They're actually an Italian Ministry of Health funded center, a standalone entity, again with 2 physician leads as well as a number of PhD and postdoc people. They're actually a center for all the Italian trainees to come in and do a simulation test similar to VESS and get involved in that. But I was more interested, obviously, in seeing the electromagnetic navigation, the sensorized tools, and sort of talking about some of these. So this here is one of the brief demonstrations that they put on here, and you can see that they actually have a 3D printed steerable sheath just there in the middle between the two. And they've created this phantom, which many of you probably have seen before now, whereby you can go ahead and navigate in this virtual environment. And you can see it's based upon this model. More interesting than that, for me in some ways, is what you can really do with, with this patient information. So this is the phantom embedded in the foam pelvis. And you can see now that they've made this 3D representation of this, and you can go ahead and manipulate catheters and wires entirely without fluoroscopy and contrast within this environment. Obviously, you can re-register if the patient moves or if you've had significant changes with the introduction of stiff aortic devices. But this is really my favorite portion of it. This is where they actually deployed a graft across it. You can see the celiac SMA and the renal arteries. And here they're using a laser fenestration technique essentially as the sort of testing paradigm. So here you can see this beautiful view which really looks inside the artery, and you can get very accurately onto the left renal orifice, which is what they're doing there. And then essentially with sensorized steerable tools and a laser fiber, they're going to go ahead and perforate through the fabric. This is the laser fiber being introduced right here. And you see it right about there. They've perforated through the fabric into the renal artery. And this is all done essentially in a virtual environment without the use of any fluoroscopy. Obviously, in a real-life setting, we would use a little bit of fluoroscopy in contrast, but this really changes how we might think about these procedures. And here you can see the wire and the catheter. So I appreciate I've gone through this relatively fast. I thought I had 10 minutes initially, and Andrew said, well, you probably should get through this in 7. So in terms of the future directions of this research, I've had the opportunity to publish with this group, as you can see on the right, and we're looking into sort of further testing paradigms for EM navigation and laser fenestration. We applied last year for the 2022 AHA Collaborative Sciences Award and got some good feedback, and we'll use that going forward for the 2023 approach. Obviously, development of novel sensorized tools, I think, is really key. That's where we're lacking a lot of decent catheters, wires, steerable sheaths, and other instruments that allow us to do the work. Finally, just a shameless plug for the VESS Travel Award. It really encourages, I think, a fantastic research collaboration that I ordinarily would not have been able to do. It fosters international recognition. Both they know a little bit about what we're doing here at the University of Missouri and vice versa, more interestingly, what they're doing at Pisa and certainly in Genoa. And I have a better understanding, I think, of the global approach to this disorder. We really are sort of somewhat limited in the United States with our access to these novel devices. And in Europe, they've been using branched and fenestrated technology for both the arch and thoracoabdominal space for decades, or a decade. So this really reinforces, I think, why we became vascular surgeons in the first place, to sort of learn about different approaches and be on the stepping stone of innovation for some of these complex treatments. Well, thank you very much for listening, and I'll take any questions.
That was actually amazing. The next award is the VESS Medtronic Research Resident Award. This award is specifically designed to help residents in training initiate projects during protected and academic developmental time that will lead to future academic potential towards extramural funding for basic or clinical science research. This award for a resident is for $12,500. The 2022 winner is Kevin Magnum from the University of Michigan for his research on regulation of JMJD3 by JAK1 STAT3 in macrophage-mediated inflammation in diabetic wound healing. Dr. Mangum.
All right, well, thank you, everybody, for allowing me the opportunity to talk, especially a big thank you to Medtronic and VESS for the award, which has been really fascinating because it's allowed me to really melt my own interests coming into the Gallagher lab in smooth muscle cell biology and phenotype with what the Gallagher lab typically studies, which is epigenetic regulation in macrophages and diabetic wound healing. So I'll be discussing my updates on how JMJD3, the histone demethylase, regulates the hypertensive gene program in smooth muscle cells to contribute to disease. So as we already saw in one of the talks from this morning's session, smooth muscle cells exhibit a marked exhibit for phenotypic plasticity to differentiate into a mature contractile smooth muscle cells, which underlies the hypertensive phenotype. However, in atherosclerosis, they shut down this gene program to this repressive state, which really allows them to migrate and grow into the blood vessel and contribute to restenotic injury. It's not advancing. Yeah, it's not advancing.
Can we have the next slide, please?
Thank you. So really the focus of the Gallagher lab is to understand how this phenotypic modulation in a variety of cell types occurs. And we know that specific enzymes, notably, sorry about, there we go. Chromatin modifying enzymes contributes to the changes in chromatin state. So here's a diagram showing that in the closed chromatin conformation, which encompasses DNA and genes that ultimately allows for transcription factors to be accessible to the chromatin. In the closed conformation, there are specific epigenetic enzymes that contribute to this, and histone methyltransferases ultimately deposit these repressive marks on histones, making transcription factors accessible to gene regions that contribute to downstream gene expression. Our lab has really focused on the study of the histone demethylase, JMJD3, which specifically demethylates histone 3 at lysine 27, basically removing the repressive methylation mark, allowing for an open chromatin state, which then allows transcription factors to bind to these regions. Notably, the regions in between each of the histones, which are accessible to transcription factors to regulate downstream gene expression, are accessible to enzymes that we call DNases. And these DNases can cleave DNA at specific segments, and then these segments can then be aligned with the entire human genome and mapped. And so these DHS or DNase hypersensitive sites can be overlaid with the human genome to identify areas of actively transcribed regions of genes. So we were really excited to see that a recent genome-wide association study that came out in Nature Genetics identified one putative SNP or single nucleotide polymorphism in the promoter of the JMJD3 gene that was associated with hypertension. And notably, it was the major or C allele that was associated with increased blood pressure. In the majority of patients with the allele frequency was 0.924. So this ultimately translates to about 10% of patients being heterozygous for this specific risk allele. So just a very brief review, genome-wide association studies, the premise of these is really identifying specific DNA sequences that vary at one or multiple base pairs that contribute with one particular phenotype or a disease state. And then the utility of these is essentially performing downstream analyses to identify the functional SNPs and containing regulatory elements that may contribute to a disease phenotype or disease state. So the first experiment that I performed was just to see if JMJD3 was enriched at smooth muscle gene promoters in order to characterize the role of JMJD3 in vascular smooth muscle cells. And you can see that there was significant enrichment above a negative control antibody for JMJD3 at multiple smooth muscle promoters. Next, I used siRNAs to knock down JMJD3 in mouse aortic smooth muscle cells and showed that JMJD3 was required for smooth muscle cell contractile gene expression. And then the right graph is showing that when you treat smooth muscle cells with the JMJD3 inhibitor, GSK4J, you have significant downregulation of contractile gene expression. This is in vivo analysis showing that in our smooth muscle specific JMJD3 deletion, and I apologize for the image quality here, you have significant downregulation of smooth muscle alpha actin and SM22 in smooth muscle cells isolated from mouse aortas in this model. Again, really suggesting that JMJD3 does play a role in regulating smooth muscle contractile gene expression. So the next part of my study was to really map out the functional single nucleotide polymorphisms within the JMJD3 gene that were regulating its expression and potentially downstream blood pressure. And so you can see the top line here is the JMJD3 gene structure, and then below that are H3K27 acetylation peaks. H3K27 is an active chromatin mark that identifies areas of open chromatin, and then these can be overlaid with subsequent other marks for open chromatin or active transcription. And one of these, which I've already mentioned, is the DNase hypersensitive site. So that's in fact what we did. And this is all publicly accessible data from the UCSC genome browser, which was a great resource for this particular project. And we were really excited to see that one of the SNPs that was identified in the GWAS study was actually located in the middle of one of these hypersensitive sites or open chromatin regions. So the first SNP you see, RS620597112. And then there was a second SNP, 744, just downstream of that, which was also in an open chromatin region that was actually in linkage disequilibrium with the first SNP. which I'll discuss here. So linkage disequilibrium means that if you have two SNPs at one particular locus, then there's a very high probability that if you have one SNP, you're going to have the other. So in this context, if you have the minor allele at SNP 620, then you're going to have the minor allele at RS744. And the reason that this is significant because in GWAS studies, which identify a cohort of DNA variants in the human genome that are associated with phenotype, as I mentioned, one or more of them can actually cause disease and regulate downstream gene expression. So my central goal here was to identify which was the functional SNP that was actually regulating JMJD3 expression. And so the approach that I used was to use PCR amplification of each of these DNase hypersensitive sites of the JMJD3 promoter, and then clone them into a luciferase reporter vector, generate an allele context of for major and minor alleles for each DHS segment. And so ultimately I had 4 different constructs that I transfected into mouse smooth muscle cells, and then 24 hours or 48 hours later measured luciferase activity. And interestingly, the major allele at the first SNP locus exhibited significantly higher transcription activity than the minor allele, and there was no allele-specific activity between the major and minor alleles at the second SNP locus. So the working hypothesis here is that JMJD3 is increased by the major allele by some allele-specific transcription mechanisms, which we're currently investigating. This ultimately removes the repressive H3K27 methylation mark, contributing to increased contractile gene expression, which leads to increased blood pressure. Future directions of this study are really identifying the in vivo role of JMJD3 in the regulation of blood pressure. So we actually have an ongoing experiment right now in measuring blood pressure in mice that do not have any JMJD3 in their smooth muscle cells. And then ultimately, we'd like to identify the allele-specific transcription factor binding events at the JMJD3 SNP locus. And again, thank you to my lab, all of my funding sources, especially the VESS Medtronic Research Award, and the organizers for the conference. And I'll take any questions.
With the incredibly high number of quality applications, the VESS Executive Council last year once again approved funding for a second Resident Research Award. The winner of the 2022 second VESS Resident Research Award is Dr. Amanda Phillips from the University of Pittsburgh for her research on improving follow-up after EVAR. Dr. Phillips will give a pre-recorded update and be available via Zoom for questions after the session.
Hi, everyone, and thank you to the VESS Executive Committee for the generous grant and the opportunity to present our progress to you today. The Clinical Practice Guidelines on Postoperative Surveillance After EVAR, published by the SVS in 2018, recommend follow-up imaging at 1 and 12 months and annual aortic imaging thereafter. It's estimated that almost half of all patients who undergo EVAR do not follow the surveillance recommendations, and approximately 40% of those patients are completely lost to follow-up. Not surprisingly, the risk of surveillance failure is even higher after an EVAR that is placed for a ruptured aneurysm. This is a phenomenon that is not unique to any provider, institution, or state, and this can be appreciated by this map that was created by Dr. Goodney's group and published in Circulation in 2020. There have been a number of studies that done to investigate how patient characteristics and other clinical risk factors associate with poor follow-up, and many of these studies have been retrospective, observational in nature, and this leaves a great opportunity to investigate how patient beliefs, attitudes, and past experiences may influence their behavior. Once we have this knowledge, we can leverage it to develop a patient-centered and sustainable intervention to improve follow-up after EVAR, which is the impetus for this study. Our first aim was to understand the barriers and facilitators to follow-up after EVAR, and this was done by utilizing semi-structured interviews to query patients, providers, and key personnel about the behaviors that are motivating follow-up. Using this rich information source, we can perform thematic analysis to identify key themes in behavior, as well as potential interventions to mitigate loss of follow-up. The second aim is to gather a comprehensive working group and use this information to develop a pen and paper prototype of an intervention that will be ready for development. Currently, we are wrapping up aim 1, which I will tell you about today. So first, we set out to develop an interview guide that would allow us to touch on everything that may contribute to follow-up without inadvertently influencing responses. But what is everything? What are all of the different domains or aspects of motivation that may influence follow-up? Thankfully, there are well-validated frameworks about motivators behind behaviors, and we chose the theoretical domains framework. It breaks down motivating factors into 14 domains, and these include knowledge, skills, social, professional role and identity, beliefs about capabilities, optimism, beliefs about consequences, reinforcement, intentions, goals, memory, attention and decision processes, environmental context and resources, social influences, and emotion and behavioral regulation. Using this as a guide, we created a consensus document of facilitators and barriers in each domain and used this to create 4 distinct interview guides. These were for patients who did and did not follow up, providers, and key personnel. We then pilot tested these guides on 1 person from each group and found them to be easy to comprehend and fit well within our 30-minute time frame. Listed are a few example questions from the patient interview guide. You can see that the questions are specific and open-ended and meant to direct the conversation to learning about specific aspects of healthcare, but are not leading. The interviewers had the freedom to probe into more detail as they saw fit, and we included a demographic survey at the end. Using our institutional database, we began by looking at patients who had an elective or emergent EVAR within the last 1 to 3 years, and this is so they had at least 1 year to have the opportunity to follow up or not follow up. From this cohort, we utilized purposeful diversity sampling, and this is to select patients for our study, which is a widely utilized technique in qualitative research to select patients that will provide the most valuable and comprehensive information relevant to our study. For example, we chose patients based on their follow-up status, whether they ruptured, their gender, race, age, travel distance, and whether they had any immediate complications with the surgery. We were able to meet all of our targets for interviews with 16 completed healthcare providers, 14 key personnel, and 25 patients, with half who had complete follow-up and half who had incomplete follow-up. In total, we interviewed 55 people. Each of the interviews has since been de-identified and transcribed, and currently there are 3 coders that have developed a codebook consisting of themes, sub-themes, and examples. And now that that codebook has been completed, they have independently coded 9 patient interviews with good inter-rater reliability. And once the interviews are coded, we can begin to work through the thematic analysis and identify the most relevant facilitators and barriers to serve as a target for intervention. And we plan to do this within the next 2 to 3 months. While we continue to work through the bulk of our project, an interesting observation has been the use of what we've called key terms from the patient interviews. Most notably, we found that nearly half of those in the complete follow-up category used the term leak or endo-leak, while less than 10% did in the incomplete follow-up group. And this finding may hint that those with complete follow-up have greater understanding about the importance of annual surveillance. Also interesting is that we found that more patients with complete follow-up were married, had a high income, and lived at home with someone else. This corroborates the finding of prior retrospective observational studies and adds face value to our study. In summary, we're about halfway done with our study, but at its conclusion, we hope to have a well-developed interview guide that can be used for future semi-structure interview studies, a clear and comprehensive qualitative methodology that's published in our peer-reviewed journals, which I truly believe can help answer some of the most difficult questions regarding social determinants of health that are so deeply intertwined with outcomes of our patients. a rich source of information regarding behavior motivations from diverse patients, providers, and key personnel from a large medical center with diverse patient population and patient care settings. And ultimately, a patient-centered and sustainable intervention to improve follow-up after EVAR and beyond. I want to thank again the VESS for their generous grant funding for this work. Thank you for your attention, and I'd like to thank my amazing team who has really helped move the project along. I hope to continue this work through my remaining time at UPMC and eventually bring it to Temple Health, where I'll be joining on as faculty this summer. Thank you.
Okay, thank you, and we'll people can reach out to Dr. Phillips offline for questions. So the final award is the VESS Boston Scientific Early Career Investigator Award. This award is specifically designed to help new vascular surgical investigators initiate projects that will lead to extramural funding for basic or clinical research. The award is for $22,500. The 2022 winner was Dr. Katherine Kehman from the Emory University School of Medicine for her research on harnessing mitochondrial bioenergetics to enhance regenerative stem cell therapies for peripheral arterial disease. Wow.
Well, thank you for the opportunity to share an update on our work. I have no disclosures. Chronic limb-threatening ischemia, despite many improvements in surgical and endovascular care, remains a significant source of morbidity. Many patients are still limited to palliative amputation. At 1 year from diagnosis, the amputation rate is estimated to be between 15 and 20%, and mortality is between 15 and 40%. The amputation risk increases fourfold with comorbid diabetes. Over half of CLTI amputees are permanently disabled after amputation. Amputation itself increases the 1 year mortality from 30 to 40% in the CLTI population. There remains a great need for improved and alternate therapeutic options. The advent of induced pluripotent stem cells, for which Gurdon and Yamanaka seen here received the 2012 Nobel Prize in Physiology or Medicine, provided a promising avenue for autologous cellular therapies, whereby a patient-specific cell, such as fibroblasts, could be reprogrammed to an induced pluripotent stem cell by adding the reprogramming factors, or Yamanaka factors as they're known, to generate an induced pluripotent stem cell. This can then be differentiated to any cell type of interest. iPS-derived endothelial cells are emerging as a future therapy for peripheral artery disease. They have certain advantages. They are scalable, infinitely renewable, and they have the capacity to be autologous. Additionally, they've been demonstrated to restore perfusion in murine models of hind limb ischemia on multiple occasions. However, iPS-derived endothelial cells over serial passages in culture lose their ability to proliferate and their mature cellular markers seen here, with the loss of EDU labeling in the top panel and the loss of CD144 and CD31 in the bottom panels. Our previous work demonstrated that this was due to an accumulation of abnormal mitochondria seen here by TEM analysis. Mitochondrial function, or bioenergetics, are tightly regulated in endothelial cells. Over 85% of ATP is produced anaerobically through glycolysis, even under oxygen-replete conditions. This anaerobic metabolism allows less reactive oxygen species production, enabling endothelial cells to use mitochondrial ROS for signaling. Additionally, it allows endothelial cells to generate energy while migrating into ischemic tissues to revascularize. In fact, angiogenesis requires the use of glycolysis. Angiogenesis is conducted primarily by progenitor-like cells called endothelial colony-forming cells that are positive for key markers CD34, CD105, and CD146. It occurs by a phenotypic switch with a leading tip endothelial cell followed by elongation with stalk cells. These TIP cells are almost exclusively glycolytic. If you block glycolysis, you cannot get a TIP cell and you cannot get angiogenesis. In order to understand the impact of mitochondrial bioenergetics on iPS-derived endothelial colony-forming cells, we performed directed differentiation of a commercial healthy iPS line Y6 using a 6-day protocol by Patch et al. That produces a mixed population, and we purified for the pan-endothelial cell marker CD144. We then performed single-cell RNA-seq analysis to understand the endothelial cell subtypes. We found there were 9 clusters representing unique subtypes. Heat map analysis of differential gene expression confirmed that these were unique subtypes. We then identified the endothelial colony-forming cells by looking for CD34 expression, which was present in multiple clusters, but predominantly in cluster number 4, which also highly expressed CD105 and CD146, confirming that these were the endothelial colony-forming cells. Gene ontology analysis of cluster 4 revealed that it was the only cluster to be enriched in angiogenesis processes and vascular development, further confirming that this was the endothelial colony-forming cell cluster. To test the effect of mitochondrial bioenergetics on this population, we then subcultured these iPS-derived endothelial cells under standard culture conditions, which is 21% oxygen, or low oxygen conditions with only 4% oxygen. We additionally used culture them with and without the drug UK5099, which is a mitochondrial pyruvate carrier inhibitor and effectively blocks mitochondrial respiration, therefore shunting all energy production to glycolysis. And then we analyzed these cells after 24 hours and after 2 weeks. The iPS endothelial cells survived all tested conditions. We found that the low oxygen conditions preserves the CD34 progenitor marker better than the standard culture conditions. All conditions in the early phase started off being CD34 positive, shown on the flow cytometry panel on the screen right here. And as we move down into the late phases, after 2 weeks, the 4% oxygen conditions retained CD34, but the late stage did not. Additionally, as I mentioned before, the iPS endothelial cells lose their key endothelial cell marker over serial passage in culture. The CD31 staining showed that the low oxygen concentrations improved retention of this marker compared to the standard culture conditions. And these cells were in fact double positive, confirming that they were endothelial progenitor cells or endothelial colony-forming cells, as they're now known. Interestingly, the low oxygen culture conditions did not have a significant effect on endothelial cell function. In our later stages, the standard and low oxygen conditions had similar nitric oxide synthase activity, similar expression of beta-galactosidase as a marker of senescence, and a similar number of surviving cells. To assess angiogenesis, we did a tubule formation assay on Matrigel. All conditions formed tubules. And when we quantified the different metrics of the tubule formation, we did not see an improvement with the low oxygen conditions in our in vitro angiogenesis. So in summary, our nascent iPS endothelial cells do contain an endothelial colony-forming cell or progenitor population. Our iPS endothelial cells do lose key cellular markers under standard culture conditions, but this is improved with low oxygen conditions. Moving forward in the future, in vivo angiogenesis capacity will need to be assessed, particularly for cells under low oxygen conditions. And with that, I'd like to thank all of my collaborators and my lab, shown here, and Boston Scientific and VESS for this award. Thank you.
Just to stay on time, we're going to keep moving forward. So I'm going to announce the 2023 award winners. First of all, before announcing this, I just wanted to thank all the committee members for the VESS Awards and Grants Committee, including Dr. Mohammed Zayed, Caitlin Hicks, Lindsay Corrupta, Matt Smeds, Kevin Tan, Sam Tyagi, and Kelly Kemp, and thank them for all their time and effort to grade all the applications for these 3 awards. Definitely, we had a lot of applications for these awards this year, and always encourage everybody in the audience to encourage their own faculty members and residents and trainees to apply, because I think these are fairly generous awards. So first of all, the first award is our Travel Award, and the winner of the 2023 VESS Travel Award is Dr. Elizabeth Genovese, who is formerly from the Medical University of South Carolina, but apparently has moved on to University of Pennsylvania over the past year. Dr. Genovese plans to visit Urbano Terme Polyclinic in Padua, Italy, to learn about their patient pathways to revascularization and their use of advanced endovascular techniques. Patient care pathways include exposure to their novel referral-to-table algorithm, multidisciplinary care team approach pathways, and follow-up protocols for patients coming from a large referral network similar to the Medical University of South Carolina. Unfortunately, Dr. Genovese is not in attendance, but we will make sure that she gets her award. Next award is the Resident Research Awards. We had 14 applications that were submitted for the Resident Research Award this year. In an effort to support our members in their research endeavors, the Executive Committee once again approved funding for a second award. Just as you've heard in this last two presentations, we had 2 awards. So the first winner is the Medtronic Resident Research Award, and that goes to Dr. Tyler Bauer from the University of Michigan for his work entitled SET-DB2 Regulates Abdominal Aortic Aneurysms. I don't believe that Dr. Bauer is here, but Dr. Katherine Gallagher from the University of Michigan is apparently going to accept his awards, which I have here. Yeah, Dr. Gallagher is going to help shuttle a couple of these awards to their recipients. So the second award is the VESS Resident Research Award. Again, we have 2 awards, and that goes to Dr. Calvin Chow from the Northwestern University for his work entitled Role of Sympathetic Nervous System Dysfunction in Abdominal Aortic Aneurysm Pathogenesis. Again, Dr. Chow is not in attendance due to travel delays. I don't know if anybody from Northwestern is in the room or in attendance to accept this award on his behalf. Anybody in the room? All right, so we'll move on. We'll ensure these awards get to the recipients. So moving on, the winner of the 2023 VESS Boston Scientific Early Career Investigator Award goes to Dr. Frank Davis from the University of Michigan for his work entitled The Role of Macrophage and Smooth Muscle Crosstalk in Abdominal Aortic Aneurysm Development. Dr. Gallagher is also going to accept this award on his behalf. And just full disclaimer, Dr. Davis, I believe, also got the Resident Research Award.
Did,
yeah,
yes,
so I think we should rename these awards the Gallagher Awards, because it seems like there's always an association with the Katherine Gallagher Lab at Michigan for these awards. So finally, we have also a Best Paper Award that we award every year, and based on the criteria for this year, all eligible papers have already been presented and graded. I'm pleased to announce that the winner of the 2023 Best Paper Award goes to Keonani Longwolf from the University of Utah for a paper on exogenous testosterone replacement therapies associated with increased risk for arterial graft infections. The recognition for this award comes with a $1,000 monetary award, and full disclaimer, I was not involved in the rating of this paper, even though I was associated with the work. So congratulations to Dr. Longwolf, and I'll make sure that she gets her award. So that concludes our awards session, and thank you for attending.